Hi, this is Eli Ocheltree of the SnoKing Beekeepers Association, and we are meeting to go over the topics that were covered at the Pacific Science Center. Sorry, I'm muting somebody. Okay, at the Washington Beekeepers Association annual 2022 conference on October 1st and 2nd of this year. And there were a lot of small talks, and some of them were super interest to beekeepers. So we're going to go through this list, I think in this order, if I got my slides in order. And it was what beekeepers really care about, I think, my pick. Sort of like recommending movies or something. Hey, and what would you want to watch more than beekeeping videos and talks, right? Swarms. Swarms are always a big topic. And I didn't try to pull pictures off this, but Ted McFall, who's a master beekeeper up at the Mountain Baker Beekeepers, he didn't just tell you how to catch swarms. He explained how they think, how they behave, what they are, then how to attract them and lure them in, including some ideas on how to capture them with a pole on a bucket. And then if you really want swarms, having that ready to answer a swarm call. He says you can fold up some of these laundry bags that have a little bit of form to them, and they collapse. The collapsible laundry bag, he says it keeps it under the car seat. He also believes in tanging. And this alone would be a great topic closer to the swarm season for a monthly meeting, or if people wanted to do it in a more informal situation, maybe. Like Jason Mayfield was talking about getting a group together for first and second year beekeepers. Then we're going to be doing the queen rearing workshop, and just about everyone has to replace the queen or does a split and has the bees raise a queen. But the next step is always, I just know I could raise queens, and we try grafting. So do it with us or with one of the other bee clubs that does it. I think West Sound may be raising queens. Some of those other ones have well-established programs. And so John Jacobs, and he's Old Sol apiaries. When you're looking for nukes to buy, you'll notice that Old Sol's on the list. But usually there's middlemen who drive down to Oregon, pick up, I don't know how many hundreds of hives, bring them to various locations. One of the reasons you need to be in touch with your local bee clubs and your local beekeepers and network as much as you can. Find out who's bringing what to you. And Caucasians is one of the types of bee that he carries. He talks about how he raises his queens. He says it's tough to do in Oregon. It's a lot like the weather here in Washington. We're still pretty far north. You have to look at the genetics. You got to have the right technique for not just for the actual physical grafting, but everything else surrounding producing a well-fed, well-reared, top quality queen. Record keeping. He says you got to put in sweat equity and have a certain amount of luck. But he says that don't worry too much about the genetics in some ways, because well-reared queens from inferior stock are known to outperform poorly reared queens from superior stock. If you can get both, great. I mean, well-reared queens from superior stock, yeah, that's probably the best. But even if you are just starting with your local queens, whatever you've got, nothing special, if you make sure they are well-fed and well taken care of from the time that egg is laid, they're in an excellent cell builder hive. And if you take the queen workshop or you work with grafting, it makes a difference. The hive that builds the cell around that queen, that egg that they are raising to be a queen, it makes a difference. He shows you a lot of things about how he does it commercially, but some of it applies to the way we do it. I don't know that I would want to make nighttime splits without locating queens, but when you're doing this in mass, commercial beekeepers do things differently. And he does 2 sessions of queen raising, I guess, late February and then in August. If I understood him, that's what he does. that queen right cell builder. How do you have a queen in there and get the bees to raise queen cells? He's got a system. And he says if those cells, good queen cells, aren't smooth, they're really nice and peanutty looking and peanut shell looking. And if when he sees little brown nubs, he knows that those people may have raised queen cells, but the conditions weren't optimal. And he's a proponent of wet grafting. And I admit, wet grafting works better for me. Oh, he happens to also sell Western frame nukes and uses 8-frame equipment, which I thought made him kind of neat for a beekeeper, a commercial scale beekeeper. Anyone who's going down to pick up bees from him, any club that's doing it, could be arranging to get Western frame nukes from him. And I know there's a market for Western frame instead of those heavy, deep frame. The nukes aren't that much heavier, but it's when you put them in the 10-frame boxes and have to work with 10-frame deep boxes. 8-frame is a solution to that, though. And apparently he uses that too. Anybody wants to interrupt me, they're welcome to, or ask a question, or put in that you actually know Old Sol. But I'm kind of running through the first part. I'm assuming people would like me to kind of go through it, kind of work in the chat and do questions and do the first, it's like the first half of the talks are really the ones I thought were important. I thought these are the ones that people are most likely to be interested in. There was one more thing about John Jacobs' queen raising. Everyone tells you to log and journal. And this is his shorthand. And I thought it was really kind of neat. If you look at his shorthand, he's got it down to he knows what these symbols mean. And most people do have a thing, a way that they mark. I like his minuses and pluses. He saw the queen, it's queen plus. If it was a virgin queen, it's V plus. I just like the way he went down it. Short on bees, S-O-B. L-O-B, lots of bees. I thought it was really kind of interesting. Oh, and if you add frames of bees, just put plus so many. And if you took them out, minus so many. That's important to remember the next time you walk up to that hive. Married? What does married mean? I have no idea what that means, what the M stands for. Okay, must be mated. Mated. Okay, somebody figures that out, let me know. Then there's a really great talk by Julia Mahood, and she's done it before, and you might be able to find her online. And she studies drones by using UAV drones, the unmanned aerial vehicle drones. This is a great excuse to go out and buy an expensive piece of equipment that you would never. I mean, it's beekeeping equipment, right? You can justify this. She flies them looking for drone congregation areas. And how does she do that? By dangling via a string. She dangles a little bit of queen pheromone, and in a little hair curler type cage. And when you go past the drones, they have incredible sense of smell, and they find it and they start chasing in the comet. And she gives you all kinds of hints. Don't just go out and do this. Don't go out and buy a $500 UAV drone, because she says you got to watch out. The drones will attack the UAV, for example. There's something wrong about it. If the string isn't long enough, they're attracted to the pheromone, then they attack the UAV. So you need her tips on how to do it. Plus, you need to have the whole excuse. And she's got great cartoons, great humor, and she thinks that drones are important. And she talks about the bees know what they're doing. The colonies only raise drones they can afford. If you're new to beekeeping, you may not have heard that some older beekeepers say they are not going to raise drones for other people's queens to mate with, and they throw out and destroy all the drone brood they come across. And she's saying, no, they know what they need. They know what makes them right. And there are other people who say this too. Others say, yeah, you can get rid of the drones, but they'll just raise that many more. because they are not comfortable without having those drones if they can afford them, if there's enough resources. She says that Google Earth Pro is free now. So check out her important tips before you try this. She's got quite a system. The reading frames talk I really liked. Sorry, I didn't put any of the pictures up here. We have to do this one, I think, for a monthly meeting. Or maybe I can work it into Journeyman. Anyway, I think this should be like required viewing for Journeyman. Much like Samuel Ramsey's video on how Varroa feed on bees and why they're such a problem. That's required viewing. You have to see that if you're a beekeeper. So he has these great slides. He goes through them one by one. He's very interactive and says, okay, now what do you see? What do you not see? Well, what if this is a summer frame? What if this is a later in the season, early in the season frame? How about if it's a larger, smaller colony? You know, not just compact brood pattern, which we talk about in beginner, but maybe you had recent treatments. I mean, if you just treat it with Formic and you don't find any eggs, that's no surprise. And there's some more talks coming up where they talk about the effect of Formic on brood. I thought that this would especially be good for anyone, not even just for yourself and a kind of a self-check, but also if you're going to start helping someone else and you're looking at a frame and you need to think about how to point out things to the person you're helping mentor. Hey, you know, things that you wouldn't think to mention and talk out loud to them. And almost everybody after the first season knows something and could help someone else a little bit, even if it's just installing the Nuker package because you've already done it once. So I think she's so good. I'm giving you some of the ones I thought were really worth spending a whole meeting on. The Pacific Northwest Survey has been done for years. Going back, I forget how many years, 10, 15, could be more years than that. And Dewey Karen at Oregon State University is in charge of it now. And he separated out the Washington data. I'm not sure they've done that before. He even had some bee clubs that were so good about reporting that he could separate out their data. If you want to see it, it's really easy. You just type this in, pnwhoneybeesurvey.com, and then look for the 2021-22 survey report. Then scroll down until you see Washington if you want to see Washington. When you see this map, you're there. It's not a large sample size, but it's an interesting discussion. He interprets the data even online, and of course he did it live at the conference, but he has comments online with this too. So he's an interesting person. I like the way he talks. And he's written a number of books too, if you recognize the name. He's actually Professor Emeritus at Oregon State University. He's been working in beekeeping and teaching beekeeping a long time. Oh, winterization and pest management. Okay, our WASBA president gave a couple of talks, and one was on pest management and one was on preparing for the winter. And some of it was really on point, like join the local clubs. Partly because you're probably sharing the same pests, the same problems, the same local conditions at the same time of the year. And you start realizing that you need a little extra feedback on how's the feeding going for someone else. How's moisture going in their hives? Whatever. Do they think they see nosema spotting? Like that. And he says you are always preparing for the winter. The winter isn't something you do one day in the fall. You are always preparing for it. And that's really true. Now, he's much more proactive than I am about winterizing. I only use a feeding stimulant if my bees just aren't taking the feed, and I'm sure they need it. He got into quite a bit the treatments for nosema, for varroa, for tracheal mites. Talked about wrapping and slatted bottom racks and the quilt boards. I don't always do all of that all the time. He's very much proactive, and he says, look at it dollar-wise. If for about $12 per hive, you could treat for pests and diseases and give your hives an edge, why not do that rather than pay hundreds for bees in the spring because they didn't make it? He does not believe in midwinter inspections. Even if it's like 65 outside, he says hives should be wrapped so as to keep the beekeepers out, so they aren't messing with their hives in the middle of winter. So okay, I'm guilty of going in if it's a really good day. And I always believe in peeking under the outer cover. I don't think he's talking about don't check the feed. He's just saying if they're wrapped, people have less temptation to start lifting boxes off. He's pro lots of top ventilation, even through the sides of the moisture shims. So this is kind of good. If you've listened to me talk about winterizing or someone else talk about winterizing, it's good to hear another person's opinion. I think people ventilate too much, actually. He recommends not overfeeding, so he thinks that's possible. I'm thinking if it's late enough in the season, it isn't, but I wasn't there, so I didn't ask him exactly what he meant by that. The Nosema treatment, the antibiotic you can give without a VFD, it didn't work for him in the feed, which I thought was really interesting because I tried that a year ago in the feed and the bees wouldn't take it either. He says next time he'll dilute it and spray drench it. Nosema is that fecal spotting. Well, it doesn't always show up as fecal spotting. That's the problem. You could have it in your bees and you could only tell by looking under a microscope at a prepared sample of your bees. And I hope that somehow local bee clubs will start having the microscopes and start being able to test for that and to do the dissections for tracheal mites, which you also, again, look under the microscope for. I did like the idea of the feeding stimulant to help get 60-pound storage if your bees are lagging. And he is very much into slatted bottom racks, which now I understand why people use them. Swarm control is one thing. Bees don't feel congested if they have a little extra space to hang out on when all the foragers come in at night. And also he was explaining that it's an air buffer because air is a poor heat conductor. And I could see that if you didn't have a lot of top ventilation. But if you have a lot of top ventilation, I don't think that it's going to work as a buffer. But it's a different opinion. So if you want to hear a different opinion on winterizing, he's very proactive. He has a lot of things. Okay, the Swedish sponge. What if we could do oxalic acid the way we do formic acid and Hopgar and Napivar? We put something in the hive and it slowly delivers for a while, and we don't have to keep going up and vaporizing our bees, changing that white powder into a icky smoke, which is actually kind of dangerous to our lungs too, all released at once. You do not want a snoot full of that. Wear respirators if you do that vaporization. So what if we could do it this way instead? Randy Oliver was working on it, and Brandon Hopkins is now coming. I don't know how much he worked on it in the past, but they're looking at a product called Varoxan, and he's showing here green control, and the orange is Apivar, which is commonly used. I don't like Apivar. It's a organic synthesized molecule. It's on my list of ones I don't like. They're not organic. They're organic in terms of chemistry. They are not organic in terms of environment. But it's performing pretty well, these first trials. And he is saying that here's some more about the trials, and he did it in hives. It's a limited number of hives. 12 of each of those conditions. And this method, basically, I call it the Swedish sponge method because that's what people have been talking a lot about. Soaking what's called a Swedish sponge in oxalic acid dissolved in glycerin. And there might be some sugar syrup in there too, in the formula. And this has been out. People have been writing about this and using a similar method in Europe for a long time. To get it in the United States and to get it for sale requires meeting all the regulations, like an EUP, an emergency use permit. So WSU and Dr. Brandon Hopkins are working on this, and they've done a run looking at adult mortality. They're doing a second run looking at toxicity, not just does it kill the bee, but is it toxic in other ways? And then the 24C is some kind of legality that you need to meet to sell this as a product in Washington, and you declare a special local need. And then he went into some other things he likes to talk about, like indoor, outdoor storage. But I thought people are waiting for this to be available. It looks like it could happen in our lifetimes. Probiotics. There was one short part of a talk from Ramesh Sagili of Oregon State University. And these are the kind of names you're going to hear again and again. So you kind of need to learn that Ramesh Sagili and Brandon Hopkins, that they're around. He looked at the value of feed probiotics, and he says that newly emerged bees have no gut bacteria. I don't know. That sounds a little odd to me because larvae eat, and then they pupate. I don't think they would get rid of the bacteria they had. I don't know. But he said that. I'm not sure he really meant it the way I heard it. But in adult bees, there's like 8 core species of bacteria, mostly anaerobic. And that makes sense. They're in the gut. They don't have access to a lot of air. That the microbiome of the bee has only really been studied for about 15 years. Now you think that's a long time, but not really. There are commercial mixes out there. People are paying for probiotics. He says really they're guesses. And they're often aerobic, dairy fermenting, like yogurt culture bacteria. And that's what I was thinking when I was reading lactobacillus, and I'm going, that sure sounds like yogurt or something to me. He talked about a study, he quoted a study by Nancy Moran, and if you need the papers and stuff, I could look at it and get them for you. Bees pass from bee to bee by the fecal oral route, okay? That's common in the animal world. He mentioned he's concerned about the antibiotics kill the gut bacteria, possibly, just like with a person. Antibiotics do have side effects, and it's possible that when we're trying to we're trying to treat for European foul brood or nosema or something, that we're damaging the gut bacteria of the bees. It's possible. And basically, he said these probiotics don't look like they could possibly work. But there are some prebiotic mixes that show some promise. He didn't have a lot definitive on that, but they're working on it. Another thing people have been waiting for a long time, people have been talking online, and Jeff brought it up again about the Paul Stamets fungi perfecti and this exudate. When the bees take it up, they take it up off the fungus. And it's the exudate from the fungus that they're licking up when they land on various mushrooms and moldy, icky stuff. And animals often know that they need to change their diet. So it seems to be that's what's happening. They've tested it in lab cages. When they say lab cages for bees, it's so many bees in little kind of like jars or bottles in a lab. And they know that bees have to have certain nutrients. They can't produce their own sterols, for example, like cholesterol and also steroid hormones. They can't produce in their own bodies the sterols. They need to get them from plants. But they use those. Once they've got them, then they can use them to make steroid hormones that they need. And polyphenols have been talked about a lot in people's diet. The colorful plate, eat a rainbow. The colorful plate is healthier for you. Diversity is better. Those items too. We're not sure exactly what it is in the fungus that makes the difference, but the trials are showing that bees are more resistant to viral infections. So it's an antiviral in some way, but it's going to be offered not as a drug for bees, not as like any kind of antiviral. It wouldn't truly be an antibiotic. But if they offered it as a drug, it would be almost impossible to ever get it approved, because there could be hundreds of compounds in the fungal exudate tincture that they're using. So instead, the FDA said, go to the Animal American Feed Control Officials. Did you ever know there was such a thing? The AAFCO. And they will run the content and safety data for a nutritional label. And then the longevity of the bee, I guess that would show toxicity or benefit, how long the bees live, and nutritional data, take both of those back to the FDA, and then they are willing to look at it as a feed additive. So people are already adding this tincture, a few people, to their bee syrup or feed, and they may actually be a legitimate. I suppose if you want to give vitamins and such to your bees, you probably can do it, but it will be legitimate and tested and approved, possibly in our lifetime. Caucasian bees. Everybody's talking about them, and the carniones and all the others. Well, Brandon Hopkins explained how the WSU collected a lot of the semen for their artificial insemination project, producing the artificially inseminated queens that they sell and are promoting. And he was telling the story of how they got accepted by the USDA to do this kind of breeding experiment. And he said they wanted them to do the breeding experiments on an island. And if you know where WSU is, and they actually had the hives in Idaho, he said there was no way that he was going to be able to do this on an island. So what he did was he said, it's like an island in a sea of wheat. And by golly, WSDA accepted that. He also in this talk talks about Caucasian honeybees in relationship to the other ecotypes and strains, and that there's 10 to 12 species of other honeybees in the world, in the tropical areas, only Apis mellifera in the temperate zone. So it's an interesting talk, not very long, in case you're interested in knowing more about Caucasian honeybees, which are known for their propolis. There's a wonderful picture there of them propolizing the inside of something or other. I guess is that the top of the frames? This was something that was interesting. I have seen this propolis dump or storage on top of the frames and not been able to figure out why it's there. When bees get into propolis collecting, and they can't, do it easily all times of the year, the sap is rising, the resins are there in, say, the spring, early summer, they have to put it someplace. I've seen this on top of not this much, but I've seen a glop of propolis sitting on top of my frames, maybe kind of mixed with wax, but it's clearly something the bees put there. That's their storage, he said. Caucasians are known for the propolis collection and use, gentleness, and white cappings, leading to the white comb honey that was really prized in the older days and is coming back in fashion. Some people are asking for comb honey nowadays, finding that beekeepers are finding that sometimes they can find a market and charge for that as a specialty product. And the darker color of carniones, not necessarily entirely black queens, but darker. And they haven't been here that long. I guess that's a long time. They came into Canada and then into the US in the 1880s before the 1922 big quarantine. And then later they were brought directly into the US. So he talks about that. He talks about where the Caucasian mountains are. They're on the Caucasus area. And for example, he went to the Republic of Georgia, that is not in the United States. So he shows pictures of these areas where they went and traveled to collect the semen for this project. I think this is the last one on this rush of talks that I was talking about that I thought were the most interesting to us. Things we're hearing about, what's going on, in the discussion groups and such. This one, though, is important because we do need to support our veterans. Healing Through Hives. This guy, Bo Grumley, and a couple of other people have a plan. They're going to connect beekeepers with resources. They're emphasizing vets, but they're not excluding non-vets. It's just that a lot of the funding is going to be for vets. They have 3 years funding, renewable, and a lot of funding possibilities. And it's not like one thing. It's so confusing. If you're interested in this, I'll have to send you the link to watch it. And Alan Woods, our WASBA president, he's a veteran, and he's very interested in this and working with it. They're not going to try to go in King and Snohomish counties so much. But Pierce, Thurston, Lewis, Whatcom, and then they will spread. And they already have properties here and there. Some of them where they could put a large number of hives, some places where vets could rent or lease a half acre of land for $50 a year or something. It was incredible, a deal to get started. And it was throughout all different areas. It was very complicated because they're talking just on the financial part, they're talking loans and grants and scholarships and subsidies. And then these larger areas where you could put in a commercial beekeeping type operation, talking pollination contracts and such. And they'll help with business planning and support you. And any education or training you need, especially they can get it paid for or subsidized or an assistance of some kind for veterans. And there's a healthy attitude to this too, and a transition back into civilian life for a lot of veterans. And the social, the networking, that's really important for success in beekeeping. And if you're a mentor and you would be willing to mentor vets, they even would kind of bring you in to how to work with the military. And I admit, I probably would not have any idea of how to deal with someone who calls me ma'am all the time. It is kind of different when you meet people who have come out of the military, are actively still in it. Then there's a whole mess of other topics, and I'm not sure people want me to talk through. Does anybody want to do questions before I go any further? Comments? Have you seen any of this? Tried any of this? I could check chat really quickly if that helps. Would that help? Yeah, people saying that they need to be the feed. The first beginners are on a schedule of feeding that you're not on the next time. Oh, the next year that you keep bees, it's that first year. And Corinne puts in there's 2 seasons in beekeeping, winter and planning for winter. Okay. I always said it was cold rain and warm rain, but yeah. Okay. So the winners are Sarah Wright gets first choice, and then Brandy Ekman gets second choice. Oh, good. Somebody put in Jason Mayfield's email. He's the guy that wants to, in the chat, that wants to kind of get first and second year beekeepers meeting together. Okay, do you want me to keep talking? I wanted to get through that first part to make sure that before 8 o'clock, anybody who has work tomorrow, has got to get the kids to bed, get the pets fed or the livestock fed or whatever, that you have a chance now. I don't mean to be overwhelming when I start talking. Everybody good? Yeah, Steve, like, no, you didn't win. Did you just get on? You're late to the show. It's in the chat. Okay. Shall I go on? It started to get more complicated, but the very first one of these less directly telling us something that's helpful was actually the most important talk, because out of the talks that are left, because Dr. Kelly Kulhanek, who is going to help WASBA by writing the Master Beekeeper academic portion of the new Master Beekeeper. And I don't know how much the Master Beekeeper is going to change. Journeyman at this point is not changing, but Master Beekeeper has been really hard to get and to even know what to do inside the state of Washington. So getting to know her is kind of important to me. And she's going to be sort of the face of WSU, the Agricultural Extension Service, in helping with beekeeper education, which they have not done in the past. So we're really hopeful this will be a good working relationship between WASBA and WSU. So, and then there's some Varroa stuff, I know, but Jennifer Hahn is interesting. She's the one that's working on the fungus that's eating, it eats Varroa mites and not bees. And she's getting strains of it that will grow inside a beehive and figuring out how to deliver it. And that would be the first true biological control we've had. You know how you can put the ladybugs in the greenhouse to eat the aphids or the praying mantises? This would be the first real true biological control. Oh, wouldn't that be wonderful? Then John Jacobs, he talks some more about a commercial operation. He did the other talk on how to get great queens. Then there's a talk by Judy Wu Smart on IPM for beekeepers. She talks pesticides and stresses on hives and maybe looking at the whole effect on the hive instead of trying to figure out exactly what's wrong. Maybe you're taking a different approach to it. Wildfires. Did you wonder if your bees were going to abscond or if it really had much of an impact on them? Ramesh Sagili looked at that. Managing Varroa mites effectively. That one, I didn't feel that was very conclusive. Ramesh Sagili's been working on the Varroa mite. Who isn't working on research with the Varroa mite, trying to solve a 35-year-old problem in the United States that doesn't seem to be getting solved? And then there was more pest management. Our president, Alan Woods, did more on that. And then there was a really detailed pesticide problem and the legalities involved and how the whole problem arose, that the water and the soil in this whole area got contaminated and the bees were suffering from the pesticides. That's a long, but it was an interesting research presentation. So let me start on these, starting with Kelly Kuhanik. And as I said, I'm kind of fascinated getting to know her because I'm hoping for a good relationship with WSU. She just became the assistant professor there, one of them, and she's going to be working with the Agriculture Extension Service. WSU is the college that ended up responsible for our agricultural extension services. They're required to do it. She's also involved in something called a national beekeeping program, collecting data from all over the United States from different states. And part of what she's doing is funded by the Washington Pollinator Task Force legislation that went through, partly because WASBA helped push. We had representatives on it. And it did some very positive things that I've talked about in previous meetings. And I can tell you what I know more if you're interested. It sounds like a lot of legalese until you actually see something in action, like an assistant professor who's actually going to help write the Master Beekeeper academic part of the WASBA course. And of course, we constantly show this graph. You must be getting tired of seeing this graph, but if once you understand it, you understand a lot of the problems. So everyone uses it. She looked at Varroa population growth and then the spread between the apiaries, like mite bombing. And she said a couple things that I thought were really perceptive. I don't know, maybe not everyone will agree with them, but I thought they were perceptive. First of all, this graph, you can find it at Honeybee Healthy Coalition, which is hbhc.com. And they actually have really good resources. They totally redid the program about a year and a half ago. And she was talking about, does it make any difference which treatment you pick? And she did a lot of data. This, as she said, is hard to read. But the next one, it says the problem we're seeing is that treatments keep the mite levels from jumping as high as they would, but they don't keep the treatment. These treatments are not keeping our mite levels below threshold in the fall, whatever we set the threshold at. So people have been saying, well, maybe they're resistant, getting resistant inside the hive. No, because the miticides still kill the bees. If you take the bees out of the hive and you put them in the cages in the labs, the miticides still kill. But we're seeing this. We know that we treat it, and we're expecting a bigger bang for our buck. We are expecting a better kill off and a better suppression of the mite population. And it gets really bad in late July. We might see it August. We start really seeing it, and it's hitting worse. It seems like it's worse every year. So is it mite bombs? Is it other beekeepers didn't treat, or and then all their workers kind of explode and go to healthier hives and move in and bring all the mites with them? Or robbing, because dearth and robbing in fall kind of go together. So she looked at this with this horizontal transmission, because it's happening. And first of all, I give her great credit for recognizing that even when we do the right thing, the mite populations are jumping in the fall. So is it, and she has a little diagram here of, is it the bees from the high mite are moving into the other lower mite colonies? She called that lower mite a receiver colony. And she set up a system of donor and receiver apiaries, and she put little cameras outside them. This technology is coming down somewhat in price. And she painted the bees. Well, actually, she got her sweet husband, who is now interested in bees because he's now working on his master's or something, and something to do with beekeeping, now that she got her doctoral and is an assistant professor. So she recruited him and maybe some other people to sit and paint dots. What you do is you scoop a bunch of bees up, you hit them with some CO2, gas them kind of, and they go, and fall kind of belly up. But she says you just kind of turn them over, and as you're doing it, you paint the dots on them before they can fly away as they're waking up. She was showing how they did that. Well, she did this, and she looked at which bees were going where. Were they going, was it the high mites that were visiting the others? Was it the low mites that were visiting the others? And it was a really interesting study. And she found out, yes, the mite load is higher in colonies that are visited by donor bees. But she actually found that it was just the fact that the receiver colonies were allowing the higher visitation. It wasn't really so much where the bee came from. So she's thinking that it's the permissiveness that's likely the important part. So maybe they're already weaker in some way, unhealthy in some way, and that's really the way to prevent this jump. I don't know, it seems awfully high. And she's thinking that robbing screens could be part of the solution. They might help interrupt this, that the more defensive a strain of bees is, the more it helps. And people have said this, the more defensive bees do better and may have lower mite loads. Not always, but anecdotally, people have said this. And so here's one of her papers that they say the jump in the varroa population is associated with visitation from non-natal bees. Means they haven't got any business being there. So that was interesting. First of all, that she even recognized that there was a problem that we all know exists, and didn't say, well, you're just not treating at all, because we are treating. Maybe not enough, but we are treating. So Ramesh Sagili went into a lot of studies. He's the next person, Oregon State University. He went into a lot of studies about, and unfortunately the audio is really poor, and he has a bit of an accent. He says it's British, but I suspect it might be something more than British. And so it's difficult to understand the audio. And the transcript didn't help either. I tried show transcript on the YouTube. He goes into an awful lot of explanation. If you're a little shaky on mite physiology and reproduction, would like to know what research is being done and how it's being done, he goes into a fair amount of detail. I don't feel that there's a lot of conclusive statements that he made. He is talking about how do you make a treatment more effective. We all know that you're supposed to treat when the hives are broodless, but I'm still not totally convinced that the bees ever go totally broodless. Although the drop into the 20s at nights the last week in my area, last week or two, I think they may have decided to go broodless. It may have happened. And then he compares effectiveness of different treatments, but the way he does it isn't necessarily the way we would give those treatments. I did like, under notes here, that he admitted that the alcohol wash sampling doesn't always work. That if they took the same sample from a hive, the different sets, they would get 50% variation in an alcohol wash, knowing that it should be the same from the same hive at the same time of year. And he admits that he knows that a gram per deep of oxalic acid powder is inadequate for vaporization. And he did finally up it. In the past, he hasn't. And so his oxalic acid vaporization always showed us it didn't do anything. He doesn't have very many hives in any experiment, but you'll notice that's true of anything that's not caged bees in a lab. Hives are expensive. So doing this kind of research is expensive. He explains some of how they do it. He has a lot of very dedicated students. I'll bet a lot of them are volunteers. I don't even know if I would do this for extra credit. They, for example, to know how many mites are actually in what brood cells at what age, they freeze the frames, and then all these wonderful young people who I guess don't have lives actually sit there and tweeze out mites and count them. Thank goodness they do that. Science, that's how science really gets done. And then from that, he can make up graphs. And the main thing is they tell you that formic acid kills an awful lot of larvae, young, old, and the eggs. And yeah, and then looking, if you treated if you treat in July and then post-treatment in August, it goes up even though you treated, which again, he's admitting what we already know is that even though we're treating, the mites are still growing. So that was interesting. He doesn't draw a lot of conclusions in a lot of what he says, and he spoke on a number of other types of research that he does. And then, oh, this is that's a wonderful picture. That picture right there, that's the varroa being eaten. This is Jennifer Hahn's mold that eats mites and not bees. And she has a time lapse of it, which is really neat. You should watch that. It just makes you sit there in the cheering section going, yeah, yes, die. Okay. Well, anyway, beekeepers get enthusiastic about odd things. And she went through it all. And this is the hopeful biological control instead of just using chemicals. And what she's doing is selecting and selecting and selecting and selecting. And she has some wonderful videos where she explains how she does this. Just like you select with plants, you keep going for the one with the biggest fruit, or it grows the tallest, or it's the bushiest, or it has the best flowers, or whatever. It's the same thing when she's doing this with this fungus. Will it grow in the hive? Will it kill the mites, etc.? And and then I jumped to oxalic acid strips. She was reporting on this too, that they're working on this. I think this is a WSU, this master student. Emily, I think, Rendelman. And that it's looking very promising. The green is here. This is the line of mites levels. I guess they're basically at treatment. point threshold that you don't want to go above, and so you're treating. And then the green is the really low mite level. If we could get this Swedish sponge, oxalic acid stabilized format for delivery. I like Swedish sponge. She also had a great diagram of Varroa destructive reproductive biology. It was really nice. And everybody uses that graph. Everyone's so tired. But she made again the point that a number of people are making is that once the mite numbers really get out of hand, it's harder to get them back down. It would be easier to keep them lower. So we need to do a better job of working on them earlier and staying on top of the monitoring. Are we up to 8 by now? Well, we're getting close. How about I absolutely stop at 8 so that people can quit and go home? But I love talking about this. This was an interesting conference. There were quite a number of topics that were better than in 2 years ago when they had the last conference, actually held a conference. Then they skipped one for COVID. And this one had a fair amount that is what is in the news and what I'm hearing beekeepers discussing. Maybe not your very first year, but you're hearing about it even in your first year. All this hope that we're going to have these solutions. This John Jacobs of Old Saul Bees down in Oregon provides nukes, which some people pick up from there and are selling up here in the state, just so you know who he is. He talked a lot about his commercial operation, and he got into the California and the almond pollination and the groundwater management problem there, and how that's going to affect commercial beekeeping. If you're going to take 500,000 acres out of food production, that's going to be an impact on pollinators. And he was talking about triple mediums. He really does use Westerns and 8 frames, and I just think that's wonderful right there. I had heard that more commercial beekeepers were starting to say, we've got to keep this liftable, manageable. And when you go to deliver a hive for pollination, it doesn't have to be 10 frames. An 8 frame is adequate. They just have to change over their beak, because for the pollination contract, the provider of the pollination hive promises there will be so many frames of brood, there will be a laying queen, there will be so many frames of stores, and it doesn't add up to 10. They don't have to put a full 10 frames in there. So actually, they can go down to 8 frames, and that means that they can do, what is that, about 25% more colonies with the same number of bees. They do need more queens, but it helps them be ready with more hives earlier, each late January, February. And then, oh, I'm jumping, I guess. I think I just jumped back to Brad. Yeah, I just want to mention that he had an interesting talk about this. If you're interested in this kind of commercial aspect, things that will affect the cost of pollination, the cost of nukes and such, there are things that will affect that. Brandon Hopkins, who I mentioned, he already gave the talk on the Caucasian bees and how WSU got into them, what they're doing with them and why. He wanted to talk about indoor storage. This is less interesting to us. The thing that was most interesting is WSU is right next to Idaho, and actually Idaho does more indoor storage. They're the number one. He ranks the top 6 here, and they're large beekeepers and that use this because it costs money and you have to be planning and you're strategizing for it. It's interesting. High CO2 levels do seem to repress mite production. There's some ins and outs of that, but I didn't think it was really of interest to us directly. Then Dr. Judy Wu Smart, she's the one that was saying, when your hives are having trouble, people often suspect pesticides, and it might be. There's a lot of effects of pesticides. Could be an outright kill, but often your hive just isn't doing well. It's hard to prove it's poisoned, And you'd have to run tests. You'd have to test the dead bees. You'd have to prove where it came from. So you don't get very much satisfaction, even if you feel it's pesticides. And when you try to say, well, the pesticides are harming my bees, they say, well, you know, a lot of your bees are surviving, they're not dying. But a bee can be not dead. LD50 is the lethal dose at which it's a dose of some pesticide at which 50% of any target animal dies. So for bees, that's the level at which 50% of the bees die. But the colony can replace those bees, but it may be damaged in a way, and the bees may not be able to maintain the colony in a way that the colony survives. So the individual LD50 of a bee is not the same as what a colony can tolerate. She studied this, a lot of bee death, maybe possibly due to pesticides, with dead bee traps. And I've got a picture of that coming up. You just simply catch what the bees push out the door and drop out in front. Oh, and actually, she actually had, so that the bees would be more likely to drop the dead bees right there. She had a system. But she also tested the effect of pesticides on just egg development by taking contaminated comb, uncontaminated comb, putting in a hive, and having the eggs develop side by side with the workers. And the contamination in the comb does affect egg development negatively. But she went through a lot. She went through very complicated talk about trying to separate the effects of stressors, all the different things that can impact your hive. And your bees aren't doing well, they're listless, they're not producing honey, the population isn't growing fast enough, all the different things that you can see and you know something's wrong. And her point was, you may not be able to pinpoint always the culprit. The way that in beginner beekeeper, when we talk about brood diseases, we say you may not know which one it is. But it really doesn't make any difference, because unless you're going to go to antibiotics, which isn't too practical for most hobbyists, unless you're going to go and actually get a VFD from a veterinarian, and you are going to actually give those antibiotics, it may not be important, because in brood disease, usually you just provide good nutrition, take out the worst looking frames, promptly remove any disease that you can, improve ventilation, your best management practices, but particularly nutrition, strengthens the colony and is often what you really end up doing. So she was pointing out that often you just want to strengthen your colony. If you can't figure out what the culprit is, she didn't use the word culprit, but that's the way I thought of it. The specific threat that you're worried about may not be the most important thing. Strengthening the colony as much as you can might be. And part of strengthening colonies that we've talked about in the past is comb replacement. And people will talk about replace your comb every 3 years. And she said, well, wait a minute. There's a benefit to replacing your comb. You're diluting the effect of the pesticides and contaminants that are there. No spores and EFB bacteria carrying over. AFB spores, which maybe they're lurking and just haven't ever shown up in an active case. She says you might decrease the risk of mite resistance. Oh, by not having constant low-level pesticides exposure for the mites all year round. Okay. And there's a problem of you've got one pesticide possibly stored in your wax, and then the foragers bring in another one, and the two can work together and actually be more lethal to your hive, damage your hive more. And she's talking about that in fresh wax is also going to help any treatments that you do. It would probably help the efficacy, the efficiency, effectiveness of those treatments. But wax is costly. I mean, that 5 to 10 pounds of wax that bees need 5 or 10 pounds of honey energy that bees need to produce a pound of wax, which they can turn around and store over 20 pounds of honey in. But And then too, when you're replacing these combs, you don't see immediate results. You're not sure what the need for replacement is. And you're only diluting, you're not getting pesticide free. So she says what she does is sort of a 3 strikes you're out system. She says one of the things that you always have at hand is your queen marker, right? And it's a different color for the year. If she sees a comb that, the bees just, it's awfully dark and the bees, I don't know, it just doesn't look really good. Maybe it's got entombed pollen in it. If something looks suspicious about it, she marks it on the top with the color that she happens to have on hand, makes an X on top. And then the next time she sees it, maybe next season, she makes another. And that way she takes out the ones that she thinks are not good. The bees are not enjoying working that. Sometimes they will avoid some comb and you wonder why. And if they're avoiding it, you see it 3 times, something's wrong 3 times. It had nocemus blot. Well, nocemus blot, then I'd probably take the comb out and throw it away right then, because that would be so much contamination. But she was saying that's the way she does it. She just monitors this way, just keeps making X's on the top. When they've got 3 X's, replace that comb. She went on and did a very long talk on a horrible pesticide contamination of water and land around hives that was killing off beehives that their university had set up. And this is how she set up some beehive traps to catch what the bees, when they come out and drop the bees. Yes, it works. And then she was weighing the hives and monitoring them. It's a very long, complex study, but it was a mess that was created and is not regulated. What they were doing was making biofuel, ethanol, from contaminated seeds that had been treated with, say, neonicotinoids. And I guess there is like tons of this at the end of the year if they produce too much. So they bring it to one facility, truck it in, and grind it up, and they're going to make a soil amendment or something out of it. But the pesticides didn't go away, the pesticides that the seeds were coated with. And under the law, it's no longer a hazardous material because the coating on a seed loses any regulation. It evades any regulation that there would have been on the pesticide itself. It was rather complex. If you want to know how complex things get, it was a good study for that. And she went into quite a bit about pesticides and how they affect sublethal and how the LD50 on an individual bee in a cage doesn't tell you how the hive's going to do, which we knew. The bee traps I thought was interesting. It was an argument for not letting grass grow around your hives so you could see if your bees are in trouble. Wildfires. There was a quick mention of wildfires, and basically there was no absconding due to it. But you would think Ramesh Sagili went through all the things that could have impacted your bees. It probably did impact the hive, but the bees seemed to be able to buffer and they didn't lose queens. That's the study he actually did. But look at all the effects that they have. We know that all that yuck all over our bees blocked up their spiracles and their tracheae. We know that. And that they wouldn't be able to smell or forage as far. That there has to be some kind of oxygen deprivation, some kind of stress on their physiology. It could have interfered with their foraging in terms of they can't see the polarized light and locate the sun. There could have been heat shock to the queens, sperm failure, and of course, all that toxic ash. Well, they didn't really find, they compared 20 hives with their queens not exposed to the fire to 20 queens that their hives were exposed to the fire. And they decided that basically the workers buffered and protected the queens. And then he talks some more about some other stuff he's doing, other projects they've been working on for a while. If anybody has a whole gram of one type of pollen, they are collecting that. And a gram is a lot in terms of pollen. They've been found that the most successful way they can do it is to capture foragers, freeze them, and tweeze off the corbiculate, the pollen pellets. Interesting study. We do need to know what our bees are actually foraging and actually using. And so we're back to the list that I thought was most important. And thank you so much for listening to all this. I probably should stop the recording so people feel free to leave and talk freely. So we are stopping